Welcome students. My name is Ms. Karen Lenisha Cabral, and today's session we will be covering up the topic on viruses Lytic cycle, that belongs to the chapter Microbes, module number 5. Coming to the outline, in this session we will deal with the definition of Lytic cycle and the process of Lytic cycle. Now what will you all learn in this particular session is the description of Lytic cycle, what exactly is Lytic cycle, followed by the mechanism of Lytic cycle, how exactly Lytic cycle takes place. Now coming to the very first thing, what is Lytic cycle? When we say Lytic cycle, it is nothing but it is a phage life that culminates with the host cell bursting and releasing virons. Now over here, you require two components. One is the virus and the other one is the host cell. Now the viruses that reproduce in this manner, wherein it affects the host cell and it bursts the host cell, those viruses are called as virulent viruses. Now the best example that is studied is the T-even phages of E. coli. Now T-even phages, when you say you have T2, T4, T6 phages, but when we talk of Lytic cycle, it is usually the T4 phage which is studied extensively onto the host cell, that is the E. coli. Now if you look over here, there is a diagram or the structure of a bacteriophage, that is a T4 bacteriophage. Now if you look closely at this particular structure, it has few components. It is a simple structure, yet it has a very great mechanism. It has a head followed by a collar, and then you have the tail structure. Now the lower portion of it consists of a tail, the sheath, and then it has some legs which are the tail fibers. And below that, if you look closely, there's something called as end plate. Now within this sheath that you see over here, inside it has a central core. Now this core is directly connected to the head. Now inside this head, you have the DNA, double-stranded DNA of the virus. Now what will happen is this virus, this entire structure is going to come and infect or inject its DNA into a host cell. Now coming to the next part, that is mechanism of Lytic cycle. Now when we talk about mechanism, it involves a few steps. So we have around 4 steps that play a role in the completion of the Lytic cycle. The first one is adsorption and penetration. Second one is synthesis of phage nucleic acid and proteins. Third, we have assemblage of phage particles. And fourth, we have the release of phage particles. We will look into each of these steps individually so that we get a better idea of what exactly happens in all of these steps, ultimately leading to an entire cycle of reproduction, that is reproduction of a virus. Now, as we all know, viruses do not reproduce on its own. It requires another host wherein it is going to infect the host in order to leave. Now coming to the first step, that is adsorption and penetration. Now like all viruses, bacteriophages do not just go and attach to any part of the host cell. Now what it will do is it will find a specific structure, or that is called as a receptor sites. Now receptors can be anything. It can be the cell wall, it can be the flagella, it can be the pili. Okay, so here it has to find a specific receptors, and only after it finds a receptor, then only it will go and attach onto the host cell. Now the T-even phages of E. coli cell wall polysaccharides or protein acts as a receptor, that is onto the host cell. So either the polysaccharide or the protein onto the host cell will behave as a receptor. Now once this happens, adsorption begins only when the tail fibers come in contact with these receptors. So once these tail fibers come and sit, if you look into this diagram, in the first one itself, you see some tail fibers attaching onto the surface. So first the bacteriophage will land onto the host cell. Ultimately, initially there'll be just two or three tail fibers that will come and sit onto the surface. Then what will happen, once the receptor site, the bacteriophage recognizes the host, then you'll have the other tail fibers that will come and attach onto the host cell. Now once this happens, in the third diagram, if you see the base plate that you see, it comes and sits onto the receptor sites, and ultimately there is a conformational change wherein the entire phage reorganizes itself. Now if you compare the second diagram and the third diagram, what happens is the tail, that is the sheath, it becomes shorter, at the same time it becomes wider. Now why this happens is basically the tube that is there within the sheath is being pushed within into the host cell wall. Now once this tube is pushed, there is a protein that is present into the base plate, that is the GP5 protein, which has an activity of a lysozyme. Now what exactly lysozyme does? It degrades the cell wall. So it will degrade this cell wall so that there is a direct entry of the central tube, and then after it has found an entrance into the host cell, the DNA from the head of the bacteriophage will then be pushed into uh, the bacteriophage will be pushed into the host cell. Now if you see the transition from landing till penetration or DNA injection, in the first one you do not have the DNA which is being and the DNA entering into the host cell, but if you look at the last diagram, you see the DNA which is being passed through the tube into the host cell. The second step will deal with the synthesis of phage nucleic acids and proteins. Now, once the DNA is inside the host cell, then what happens? Now, the host cell itself has its own genome. So, the host DNA behaves or serves as a template for the formation of mRNA. Now this mRNA is called as an early mRNA. Now why is it called early mRNA? For a simple reason, because it is formed even before the viral genome is made. Now what is the function of the early mRNA? The early mRNA is then going to form some proteins along with some enzymes. Okay, so uh, from transcription there will be translation that happens. And now why are these proteins and enzymes made? For a simple reason, because these enzymes will now take over or hijack the DNA machinery of the host to form its own viral components. At the same time, once this is done, once the machinery of the host is hijacked, now what it will do is the DNA will act as a template for the viral genome, and the viral genome is made in using the host machinery. So once that is done, once the DNA is made, so then what will happen, this one of the template will behave will act as a late mRNA. Now later mRNA is used for the next step, wherein the structure of the bacteriophage will be made. So if you see in this diagram, you have the DNA. So when we look at the first DNA that you see, it is the host genome which helps in the formation of mRNA, followed by early mRNA, followed by the formation of protein, wherein the protein will destroy the genome of the host and then form its own viral genome. And after the viral genome is formed, you have the formation of the late mRNA, which will be utilized to be for the formation of the other parts of the bacteriophage. Coming to the third step, then we have the assembly of phage particles. Now, assembly of phage particle is a pretty complex process, but it is self-assemblage with the help of some proteins and the host factors. Now, because for assemblage of phage particles, we have the late mRNA which comes into picture over here, wherein the late mRNA synthesizes three kinds of proteins, and all three are very, very important. The first one being the phage structural protein, followed by proteins that help in phage assemblage but without acting into the viron. And you have the third one, which is the proteins involved in cell lysis and phage release. Now, from these three proteins, you will have the first two which will play a role in this particular sequence, whereas the third one will play a role in the next step. Now, when you say structure, now all the proteins, all these three proteins will be made simultaneously, but later on they will be utilized in their own fields. Now, how exactly it happens? For the first, if you look at the diagram over here, you have different parts made separately. So the first formed part is the base plate protein. So first you have the formation of the base plate. Once the base plate is formed, onto the base plate you have the formation of the central tube. This is the tube through which you will have the DNA which will pass through. After the tube is being formed, then around the tube you will have the sheath which is formed. So that is your protein sheath. And then you have the next part, you have the prohead, wherein you have the head which is being made with the help of an extra or external protein that is called as scaffolding proteins. Now these scaffolding proteins are then destroyed once the prohead is constructed. So after the prohead is constructed, you have a portal protein which sits at the base of the prohead, and that will help in the assemblage between the assemblage of the prohead and the tail. After this particular component is done, at the last you have the packaging of the DNA. Now the DNA is packed with the help of a protein that is peccasomes. And then we have the last step, that is the release of phage particles. So the last step is very simple. Once everything is packed, once the assemblage is done, the host cell will have all the DNA. There will be around 150 virus particles that will be accumulated within, and then there will be a release of these particles. So you have two types of proteins which are involved. One is the T4 lysozyme and you have the Holin proteins. So the T4 lysozyme is basically it is going to destroy the peptidoglycan, that is the wall layer, and then you have the Holin which creates a hole in the plasma membrane, therefore allowing the lysozyme to move smoothly towards the cell wall and disrupt it so that the bacteriophages are released. Now this is on a whole the life cycle that is drawn. These are all the steps that I just explained, wherein you have the infection, the injection, the penetration, then you have the formation of the phage particles and the viral genome, and then you have packaging, then the viral components which are made separately, then the assemblage of it, and then we have the release of it. Thank you.